So I presented data from the 11 study. This was a very large study performed all around the UK. And within the study, we were able to compare of a quadruplet combination as induction therapy for newly diagnosed myeloma patients compared to a triplet approach. So the quadruplet included the combination of carfilzomib, cyclophosphamide, lenalidomide, and dexamethasone. And we were able to compare outcomes for this quadruplet within the study to those for patients who'd received the triplet of cyclophosphamide, lenalidomide, and dexamethasone. So essentially with or without the carfilzomib included. So this was a phase 3 trial for newly diagnosed myeloma patients who were considered fit to undergo autologous stem cell transplant. So the study initially compared two different triplets, one comparing thalidomide and one lenalidomide. And then the study was adapted when the second generation proteasome inhibitor carfilzomib became available to look at the combination of carfilzomib alongside the lenalidomide triplet. Patients were randomized, therefore, between those two approaches, between the quadruplet or one of the triplet as induction therapy. So the results for progression-free survival for this comparison have been previously presented and showed a significant improvement in progression-free survival with the addition of carfilzomib to the triplet compared to those who didn't receive the carfilzomib alongside the cyclophosphamide, len, and dex. What we presented at ASH this year was the overall survival data for the study for this comparison after we have done almost 10 years of follow-up of the patients within the trial. What we found was a significant improvement for overall survival for those patients who received the quadruplet combination compared to the triplet. So I think our study really showed the importance of combining a proteasome inhibitor and immunomodulatory drug up front. We have seen that that approach was better than using a response-adapted approach, so using only a proteasome inhibitor for those patients who had a less deep response to their initial IMiD induction triplet. What's important is that as we go forward, we have now seen ongoing results from other studies that have shown the addition of an anti-CD38 antibody even into that mix on top of the proteasome inhibitor and IMiD combination seems to also show additional benefit and is now standard of care worldwide. But I think really importantly, what we showed is that really long-term follow-up of large academic trials enables us to read out overall survival data. And it really is necessary to follow up these studies for a long time to get that kind of granularity of data and be able to show these incremental improvements in overall survival that we've seen as treatments for myeloma have evolved over time.